Well, since chromatography is the youngest, obviously, of the disciplines, I think, when it comes historically and all that, I come as the last speaker here. The important thing about chromatography is right now it's about 110 years old exactly this year, so it does have interesting history. And when I think about it, you know, I think the first time I heard the word chromatography is actually more than 50 years ago, and I kind of loved the concept, and it has been a good part of the things that that I have been doing. Obviously, 75 years anniversary tells us that it started in 1938. So 1938 was not a particularly good year. This is when Europe was pretty much gearing for the war, and the things were influenced by that quite a bit. Nevertheless, a lot of things did happen at that time. Chromatography was known to very few people. So when I got involved in the field, I really have been able to witness what tremendous changes from about 1960s, 1950s, 1960s or so happened to the analytical separation science. The resolution, basically, people were talking about the mixture components with you know, just a few components in it, and today we see up to thousands of peaks in single analytical run, speed of analysis from days to hours to minutes to seconds, and even down to milliseconds, as I'll show you later. Sensitivity improvements due to the availability of the detectors, you know, change enormously as well. And so the information content, for example, in combination with mass spectrometry and the computers that we have today, the computational techniques and all that, to rapidly not only to separate but identify the number of components in the mixtures is tremendous as well. And a lot of things happen also in terms of miniaturization. Millimeters to micrometers, even nanometer dimensions, as people work today because of the microfabrication, the microfluidic manipulation and all that, so allows people to work on very small biological objects, like, for example, the single cell. Oops, that's not the one. As you remember, many of you remember that actually the father of chromatography is MIlos V. He was a Russian. The Cvet means color, and I think this comes as a misnomer that has been with us for a long period of time. So he was well aware that you can separate chromatographically substances from each other, whether they are colored or not, but he was kind of ticked off that some of his contemporaries, in fact, didn't like his work. He was kind of a maverick among the scientists, so he said, well, why don't I actually name this particular method for myself? So Cvet means color, chromatos in Greek means color. So that happened at the beginning of the previous century. And of course, the most important developments in chromatography date to Archer Martin's work on partition chromatography, which took place in the early 1940s in forms of a paper chromatography, and then later on suggesting gas-liquid chromatography, which sort of is shown on the lower part with the chromatogram obtained in rather primitive condition, nevertheless showing that, it is possible to quantitatively measure. And I think this is really where chromatography and analytical chemistry started to merge more significantly. Before then, it was a sort of a method mostly for isolation rather than measurements, or very sort of rough type of measurements at the best. So Martin received the Nobel Prize in 1952 for the number of things that he has done related to the partition chromatography. And mostly paper chromatography. Interestingly enough, there were some other aspects of their work. They came with the first type of theory known as theoretical plate theory. That term has been with us ever since that time. In the era of electrophoresis, the things were in many ways, somewhat similar. These were not quantitative type of procedures. And of course, the person who is known for the discovery of electrophoresis is Arne Tiselius at Uppsala University in Sweden. And he also received Nobel Prize. Also, his scientific father received Nobel Prize. That was Tess Werberg for ultracentrifuge, the same university, so was the whole tradition there in Sweden. And he influenced and had a number of his students who were quite active in the field of chromatography. The person where my sort of personal story started with is actually less known to most of you. It was Jaroslav Janákin in Czechoslovakia, then where I was raised, I was born and raised, and he sort of his personality attracted me away from biochemistry. My original training was in biochemistry at the University of Brno, but I wanted to do chromatography. I actually did chromatography in primitive forms and did a little bit of research in the then not very common technique that was thin layer chromatography, which was just so much faster than the paper chromatography, which the biochemists practiced all the time. It was the only form of chromatography, column chromatography as well. And so he, as a young chemical engineer, was in charge of running the analytical lab in a chemical industry in Czechoslovakia, and he needed to measure the gases, actually the concentration of different gases in a variety of products. And at that time, the sort of established methodology for doing that was the low temperature distillation. And so in 1948, when he got his job or so, there was an embargo imposed on Czechoslovakia because there was a coup. The Czechoslovakia came under the communist government, and there was the embargo on the apparatus that was produced in the United States at that time. And so he had to come up with alternatives. And I was searching quite a bit about finding, some of the original work and all that. It was early 1950s or so when that procedure was patented. And so fortuitously, this thing came just very recently out of Brazil. Somebody wrote an article about this important type of development. And what you see there is one of the first gas chromatographs that was produced in the world, where actually you produce carbon dioxide by the chemical reaction, well-known chemical reaction. Then you regulate the pressure and volumetric flow rate into the chromatographic column. The adsorption principle was used for the gases, and then you introduce the sample into the column, which is 7, and of course the description there is in Portuguese, which you and I probably won't read, but it's similar enough to Latin, so we can look at that. And then in the end, you don't have the usual detector, but you measure in a microburette, so you measure the changes, the volumetric changes, and before and then, of course, the carbon dioxide is then absorbed totally. The carbon dioxide that was used as the carrier gas. So this became a patented procedure and actually was used all the way up to, in not this primitive form, but rather instrumental form, throughout Europe in quite a bit into the early 1960s or so. So that made Professor Ayala kind of known to the community, and I was very much attracted to the institute that he established there. And of course my my biochemistry teachers were kind of horrified that I would leave the field for something like analytical chemistry. So I was very much influenced by these, what I would consider the first landmark contribution to the GC decade, starting with Martin and James' description of the gas-liquid chromatography. And as I said, this becomes an analytical method when it clearly, this became a precursor to today's instruments. Samples can be analyzed online, various detectors could be used, the methods are automatable. Of course, the method needed the theory and the description, and that here comes the first important contribution of the American academician, and that's the Giddings and Eyring. It's much less known that the well-known Van Deemter theory, which came out of the Netherlands, and then of course the theory of the the capillary column that whose father was Marcel Golay, who was working as a consultant for Perkin-Elmer. Obviously at that time people were going down with the diameter of the column as A.J.P. Martin had predicted, more or less without any experimental evidence. He said we should be able to work from milligrams down to microgram scale. Well, that was a big revolutionary step at that time. That would imply we decrease the diameter of the columns correspondingly, and this, of course, will have advantages of their own. Then fortuitously, the first ionization detectors were also reported around 1958. And also people started to do, in however primitive form, the first GC-MS combination, because these two instruments combine extremely effectively in a way that GC provided the separation, mass spectrometry provided the identification. Before then, the gas chromatography, no matter how powerful in separation number of components, just produce you the dozens and dozens of peaks, and you may not have a faintest idea what these peaks are. And some of the most important, that was also the hero of us, was Dennis Desti, a chemist at British Petroleum, who in fact constructed some of the first viable capillaries. So Marcel Golay, the idea and the theory, the columns that he presented at that time actually didn't look very impressive, but it was Desti who in fact put the capillary GC to the audience, basically to say this is really something you should be doing if you care about the separation of complex mixtures. However, he did that only in sort of for petroleum type of sort of samples. So here it comes. In the middle of difficulty lies opportunity, and I am pretty sure that may have been on the mind of Albert Einstein when he had to leave Germany and came to this country to become as influential individual as possible. It is not easy for a person to actually leave one's home country and just face face the world. My situation was not what I would consider a desperate one, but nevertheless, when I was coming to the Stockholm train station with $22 in my pocket or so in January 1968, you know, again, I had no idea what was ahead of me. At that time, I was planning to come to Sweden and learn about GCMS, and then go back to my institute in Czechoslovakia to start another section there that would be involved with this technique. What I didn't know at that time is how to make glass capillary columns, and also how to make them effectively working for a variety of samples. So this is the machine, the capillary glass drawing machine that Desti kind of described in 1959. Now, in 1965, 66 or so, one of my tasks was to actually reproduce this type of machine in our institute in Brno, Czechoslovakia, and to basically get the capillary columns. And so I burned a lot of my fingers and all that while I was doing that kind of work, because the hot glass looks to you optically about the same as the cold glass. But I had that, and this for a number of years was actually things that inspired me, and interestingly enough inspired some of my students at Indiana University in early stages of their career, the folks like Milton Lee and Jim Jorgensen and Mike McConnell and Jerry Rhodes, just to mention a few. They were the people who were inspired what the capillary columns could do. Of course, it was not as easy to get to the stage where you can make highly efficient columns. That means the task after you have drawn the capillaries is how to put the stationary phase inside and how to put it there as a very homogeneous, uniform film. And of course, what was against us was the contact angles that most of the organic liquids, including polymers, exhibit on the surface of glass, as shown up there. So you know how to beat that problem and how to basically make the columns to behave so you deposit sub-micrometer layers in a reproducible fashion. So there was lots of modification of surfaces that later on I had no idea actually when I was still in Czechoslovakia how important that work would become. But fortunately enough in Sweden people were very interested in having high resolution type of columns. So what we were doing is treating basically the surface of the glass capillaries with the variety of corrosive agents, HCl. Then we have actually a procedure by which we could sort of create whiskers on the wall, the micro kind of modification of the surface that the stationary phase would lie and behave as a stable film in it. So as Denis Desti by himself referred to, is a coating capillaries is a black magic, bash it through and hope type of method. So he produced some good columns, but it was not easy to do. Here is just one of the things that we later on only learned what we were doing to the surface. I already mentioned the whiskers of the silica type of materials. You'd be soaked with stationary phase. GCMS is where what I was supposed to learn when I went to Sweden. And of course, they were some of the pioneers, pioneering papers in the 1960s. Mid-1960s or so, that's where all the battles were on. And in fact, you know, last two papers, these were highly competing laboratories, the one of Klaus Biemann at MIT and Ragnar Rihage in Stockholm. So I joined the Rihage's laboratory and work in there. And of course, the whole thing is which of the so-called molecule separators that were used for separating of carrier gas from the molecules of interest could serve best as the interface for between GC and MS. So this is the Rihage type of separator, where you could see in the jet there is a separation. Most of the smaller molecules are removed. That is the carrier gas, helium typically being used, and then most of the somewhat heavier molecules, because they have a different kinetic energy, they come all at the same speed then into this interface, then continue to the mass spectrometer. So the yield of these things was about 30% or so. So that was this type which was pursued commercially in Sweden, and it was another type, the one of Beeman, known as Beeman-Watson separator. This is totally history today. Was the one which would allow through the porous glass, in fact, the carrier gas to be removed. And of course, you can guess which of the two concepts would be the winning one. This was a slow process. You were losing most of your sample and all that. So. But there was a fairly brutal, at that time, sort of a battle between those laboratories. I could see the very severe competition, as the first time also met Klaus Beeman in Sweden at that time. My boss, Ragnar Rihage, on the left side sitting there, and then the guy who is standing there is actually Chuck Sweeney from Michigan State University. Well, the fact that I'm standing here and all that I owe in a great part to Chuck Sweeney, because when he came to Sweden at the time when Czechoslovakia was invaded by the Soviet Union, so there were tanks rolling in Prague, and he said, well, you're not going back there, are you? Would you be interested in coming to America? And I said, yes, I have to think about it. And I've been thinking too long a time about it. So by the time his offer was off, he hired for his postdoc position somebody else. But he said, I would recommend you to anyone else in the U.S. if you want me to as introduction also. So I had correspondence with him. I was interested in working with Carl Giddings, and I was interested in working with Al Zlatkis. So those were the two names. Giddings didn't have a fellowship, Zlatkis did. But before then, I wanted to say that the work that I did in Sweden was on capillary columns, and in fact the capillary column techniques that I learned in Czechoslovakia became in my new environment sort of important in able to get the recognition with my new peers. And so I remember some of that work was I was trying to send it to analytical chemistry. It just came back without a review saying not of interest to any wider readership of analytical chemistry. So we had to publish these things in Chromatography journal. It was kind of interesting to see that these were some of the first viable capillary GC-MS separations. Here is the column. This is the separation of tobacco smoke. About 500,000 theoretical plates are in that column. We injected that at very low temperature, so that was the success of being able to analyze this very complex mixture. Al Zlatkis was extremely important for my development, and in fact, I guess is a founder of this American sort of dynasty, or so I might call it, of the number of people who have been successful in the field. And of course, he influenced many fields with the laboratory, which was quite small, at small university. And what he really provided for me was excellent environment to do research in his laboratory. I could do just about anything if it wasn't terribly expensive. So, he allowed me to continue my work. He also introduced me other things. I worked on GC detectors, supercritical fluid chromatography. HPLC was just about making its way into more laboratories. That was a brand new technique then. And he let me teach a part of his graduate course in chromatography and participate in writing grant proposals. We never got any of those grant proposals funded, but whatever I learned, whatever I learned was very useful for me when I got into the academic position. And, the University of Houston, it was not a big place, and the chemistry department was not a big place, but just the whole number of these world-class leaders, including Archer Martin, Ernst Bayer, Gilad, the number who was who in chromatography was visiting Houston. And also because Al-Zatkis ran this very successful symposium series, Advances in Chromatography. So, I met there a number of those famous people that I could only admire by reading before. And so he did a lot for me, and he was emphatic that I apply for academic jobs. I said, come on, I came from nowhere, at the site of the water and all that. The people who are in academic jobs are people who get their degrees from, respectable American universities and all that. I don't have any chance. He said, no, America is different, Milos. So, apply for the job. So in order to please him, I applied for 5. And I got 3 interviews out of this, which wasn't too shabby. And, during my first interview, I sort of I said, gee, this is something I really would like if I could get this kind of a job, be on your own and be able to train other people and all that. Because otherwise I had lined up a couple of industrial jobs on the West Coast. So, I was ready to go to industry. What he didn't tell me about, was, one should negotiate in the academic interviews. Space, equipment, teaching. I was totally ignorant about those things, coming from Europe. So, when the chair of a department would tell me, well, your salary would be such and such, I say, yes, sir, so there was no discussion. Space, I was put into a 4-man lab, so how, and squeezed on both sides by the laboratories that were not going to move. So, within about 2 or 3 years of my existence at Indiana University, I had to be on 2 different floors. He didn't tell me about the scientific dynasties, how important that was also in a way. Well, what are the dynasties? I certainly knew that there was a whole bunch of, Klaus Biemann had his own dynasty, all the famous mass spectrometers, mass spectrometrists that he trained, in his laboratory. And so that's one of them. Howard Malmsted, it's another dynasty there. Gary is a part of it, and so are many other people who graduated from University of Illinois and all. So academic politics in general, I was pretty ignorant about, so I had to learn. The other things that I learned in Houston is that, we have all these different people visiting and all that, but not too many people who were in the academic laboratories in the U.S. But one person that I was sort of picking up some stuff in the lab, and I was supposed to work on all kinds of bizarre type of detectors, was Jim Lovelock. This is a great picture of him. So he was a frequent visitor. So I inherited, so to speak, some of the project after him. The only thing that Jim was not very good at was making any documentation on any of the things. No lab notes, no nothing. He was known just, there comes the idea, came to the lab, the things worked. He's a genius. And of course, well recognized for all the different things he has done. What I find interesting in this symposium here, that we see all these people at pictures then being taken, pictures being taken, they smoke pipe, right? No more of that now. So the state of academic separation science, and I underline academic science in America in the late 1960s. Industry was extremely knowledgeable. Of course, the gas chromatographs and then later on the liquid chromatographs, keep doing this, that they were in existence, but they were only exceptionally a few of the university professors that were active in the field of chromatography. Zlatkis was one of them at small university. Carger at Northeastern University was just beginning. Harold McNair, Juve at Arizona State. Buck Rogers at that place up north was there. Csaba Horváth, this is interesting. I mean, the pioneer of HPLC. He had to be like 50 years old before he got tenure job at Yale. I think that's just disgraceful. The European countries, I think, were obviously far ahead of the U.S. Even small European countries like Netherlands, like Sweden, and not to speak about UK. I mean, there was lots of activities in the field of chromatography. So not surprisingly, most consultants to the U.S. instrument companies in that field were foreigners. So a question arises, was chromatography or separation science in general considered then a part of analytical chemistry? I'm pleased to see that we are part of this quadrangle now. But certainly 1970s were kind of lonely when I would go to MUAC meetings and analytical meetings and all. So I was kind of an oddball there for a little while. And during the interview, the same thing. One person that I admire greatly, of course, a great American academician, what I would call the prophet in separation science, is Carl Giddings. And these are some of the things that are due to him, the 3 books that he has written. I think The Dynamics of Chromatography, is a masterpiece that my students and everybody had to be exposed to it as much. I mean, I read it religiously. Then there is the other book that he published later, which is being in wide circulation still today. And then what most people don't know is that he was an explorer. He went to South America. In fact, it's described in this fascinating book, Demon River Apurimac, that he has discovered some of the origins of the Amazons in Peru in enormously adventurous type of mission there. And so I did not know him until about mid-70s, when actually that was when he was doing some of these explorations in Peru. He just took probably 3 years off or so. And we became very, very good friends, and I miss him very much that he's not around. Well, he was a prophet. I mean, obviously he had in some of his publication, predicted so many important things that happened there. The ultimate column should produce 1 million plates or so, do so quickly. Well, that has been a continuous dream, but the dream is becoming a lot more the reality in the recent years. High pressures, he sort of emphasized that well ahead of the time of the others. So it comes from actually his lectures in 1964. And the particles itself has to be very small, but has to have internal surface or the most with the thin porous skin. So, these things became reality also much later than he predicted them. Well, Indiana University has been my academic one and only academic home for over 40 years. Most of these years, a majority of them were very good years. They were not so good years as well, but that's sort of chemical noise when it comes to it. What attracted me to Indiana University was excellent faculty. There was people who were writing very influential books, textbooks and all that. Biochemistry was a strong component at that time of the chemistry department, having Gary Heafy as a colleague. I mean, nothing was wrong, with all the others, but, from all the interviews I had, I remember most vividly the one that I had one-to-one with Gary, and I was very impressed with his insights, and I really wanted him to be my colleague. Bloomington is a beautiful place. It's much prettier than the rest of the Midwest. And I had some very interesting trip there, and I won't go into any detail of that. I said, oh, that's kind of interesting place. So I turned down the other offers and came to Indiana University. And bunch of students I had, I never regretted that decision. You see this is probably the first research group I had. There, Mike McConnell sitting next to me. Believe it or not, I had some hair at that time. So I'm in the middle there, and behind me is Milton Lee, whose sort of the haircut was a little bit different than it is today. So that was a remarkable group of people, and many other students that were there. There is our first commercial GCMS, and we didn't have enough money for data acquisition system. So what you see in the back sitting there is actually Texas Instrument homemade type of a system that was working for us for a little while. I remember recording spectra still on oscillographic papers. That's, the times have really changed in mass spectrometry. But we were producing very nice type of results, and this is actually coming from a body fluid type of analysis, and I won't go into the details about what we were doing and how and all that. You could see that of course something like human urine does have lots of components in it, and we were busy identifying, we were busy at that time correlating the state of health. At that time we were working mostly on diabetes related type of problems. So these were some of the precursors, I could say, of what people today fondly called metabolomics. So that was my biochemistry background that has been sort of combined with my analytical inclinations. At that time also there was the space age applications, which was mostly done in the US. Of course, example of collaboration of in the federal laboratories, NASA, industry, academic researchers. So there was supposed to be lunar gas chromatograph, and in fact Sandy Lipsky and Jim Lovelock and others were involved with that. It never materialized for obvious reasons, that the samples were brought back through the Apollo mission and analyzed a number of laboratories. I was always fascinated by this world. I never thought, though, that I would have anything to do with the space exploration. However, there came the Viking project and the GCMS to analyze Martian soil for the presence of organic compounds. That was about my second year at Indiana University, when actually John Hayes was a colleague who was connected to the Beeman's group, and NASA. Beeman was in charge of the science team on the Viking project, and so they came up with this GCMS exploration. They needed really a good GC column, and they didn't have it. So, at the beginning, they wanted me to look at some of the candidate columns, and of course, I was kind of more or less saying, yeah, okay, I can pay my students for some of the money that they were offering us, and then more and more they were saying, well, would you have a better solution? So we came with the better solution, and in fact, our GC column was sent to Mars twice, 2 different sites, and we published a paper on performance of that particular column and what it did. However, I did not take any part in the interpretation of the results. That is sort of still an interesting point. But here I was. This is again was to me America at its best, when I was accepted to the group of people, again as an outsider or so, and they were all these famous people you could see on that previous slide who participated in Leslie Orgel, Harold Urey, Carl Sagan part of that group, and that. So I was really flattered by this. The interpretation, there's a problem with the interpretation, has been recently pointed out, and this is contrary to 30 years of perceived wisdom. Viking did detect organic materials. It's like a 30 years old cold case suddenly solved with new facts. And that's what Chris McKay of NASA is claiming. So there is now curiosity on Mars, and I'm anxiously, hoping that they will finally put this particular thing, to rest. Is there, or has there been life on the planet of Mars? The culprit here in the experiment that no one counted on was that there is a lot of perchlorate on the surface of Mars. So what was mostly detected were chlorinated, actually, hydrocarbons, but high parts per million. It cannot be explained by terrestrial contamination. So that story is still, to be solved, and of course we will see how that happens. This comes from Bloomington Herald Times. Not the last year when Curiosity landed on Mars. Got out to make room for new inventories. There is the Viking sitting someplace there saying 76 low miles. Didn't move anywhere. So that was an interesting kind of sidelight of my my sort of time at Indiana University. There were many key important contributions from industry, and in fact, I was I was glad to be part of it. In the 1970s, I was consulting for PerkinElmer. So these were some of the things. The few silica columns reported then earlier from industry. LCMS came into existence and all that. Our other contributions have been in making, this came in the post-tenure era, because I wanted to miniaturize the things in the condense phase as much as we did in the gas phase. And so those are just some of the key papers. And after tenure, you say, well, I can do any damn thing I want to do, right? Well, it's not really like that, but there was lots more freedom. And so the 1980s were interesting times. So we designed the capillary LC column. So this is the first capillary column that was designed. It's just some details of that. And the chromatograms, which were about 85,000 theoretical plates in 1978, not so shabby, I would say, by today's standards. So I thought, all the leaders were going to praise me, and that's what everybody wants, a young scientist wants. Well, instead, there was lots of controversy, lots of battles about it. Why do you do such a stupid thing? Make columns smaller. This HPLC works so well. This is LC1509. It's old science, isn't it? Yeah. And until I realized that this is actually what happens very often in science, as this famous biologist says, there is normal process of acceptance. First they say it's worthless nonsense, then later on, yeah, okay. But, at the end they say, yeah, I told you so. Yeah, you should have done that. That's good. We all do it. So attractive features of very small columns as high efficiency, speed of analysis, improvement in detection techniques. And this is old slides that I found here. Easy hyphenation, LCMS, of course, it took another decade or more than a decade to do that. The small sample analysis down to the level of maybe individual biological cells and all that. Solvent economy, environmental benefits. People were laughing about it, way back, and of course it is an issue. So the significant process in analytical separation is the 1980s. So capillary GC-GCMS became very, very sort of readily available thanks to many laboratories. Enormous expansion of HPLC in terms of new columns, but it was good to see, the new people coming into the field in the U.S. specifically. Fred Regnier, who was biochemist originally like myself at Purdue University, Pete Carr, University of Minnesota. He left electrochemistry and started to do pretty much full-time work in chromatography. George Guillochon, a French scientist who was well established there, moving to the U.S. Industry showed tremendous interest, and enantiomeric separation became of a big issue and needed to have the HPLC type of solutions. Capillary SFC, capillary LC, like a capillary electrophoresis under extensive development. I need about 5 minutes, Dennis. And what was interesting to see that it drew both established scientists, spectroscopists, Ed Young, Dick Zare, Barry Karger, others, Norm Dovicki, Andy Ewing, Sweedler, Bob Kennedy. Those are the people who actually were starting during that period. Capillary electrophoresis, very quickly. There's a lot of people who were interested in this particular subject, but they were not right at the point. And so when Jorgensen and Lukács published this revolutionary paper in 1981, Jim Jorgensen is my student, and is the study that really revolutionized the field. And that is to go to very small dimensions where you can dissipate the heat because the efficiency of separations in capillary electrophoresis is proportional to the voltage, and they're indirectly proportional to the diffusion coefficient. So larger the molecules is the better. And this is from the meeting that I organized with Professor Ishii from Japan. This was a US-Japanese seminar. About 50 people was the maximum that we could have there. And what's interesting is now you could see the people who are not chromatography people who are in that audience. The middle there is Jegenian, the mass spectrometrist. There is Willy Simon, who was already mentioned here. This electrochemist wanted to have tiny electrode working together with the small capillary type of columns. Ed Young, Joel Harris. You will recognize at least some of the people there. Jim Jorgensen sitting in front, they were all younger. So the instrumentation that we have to get more heavily involved in the 1980s sort of had us to learn a lot from the likes of Ed Young, from the likes of Dick Zare and all that. So these instruments then, our laboratory started to look like the one of Gary Iffy almost, you know, with all the optical benches and all that, because these things were not readily available. This is a good one. I always appreciate this chromatogram that was delivered to me by FedEx. There was a slide at about 11 o'clock. At 2 o'clock in the afternoon, I had a lecture, because all these people who were trying to combine laser with capillary columns or so, they didn't have any applications. They were just showing, you know, here is a riboflavin, or here's a one peak kind of thing. So you do chromatography to separate one or two peaks. Well, no, you need to see the whole profile. So when finally these results came, I was so happy to show it at the symposium on HPLC. There is the whole area of this very small type of micromanipulation type of things that became possible because of small columns. And of course, A.J.P. Martin himself made this projection. He says appetite of a chemist to work on small scale will grow, will analyze and experiment on single cells. Well, that was like science fiction in 1962. I think one of the best examples of all times is the work of my dear friend Mark Whiteman, together with Jim Jorgensen. Mark, meanwhile, moved to North Carolina, unfortunately. This is a single mammalian cell, and what we see there with electrochemical detector coupled to a capillary then is the usual neurotransmitters and the sort of things that Mark has been interested in. A breakthrough for us came with actually going to small columns packed with small particles. And this was the work of Karl-Erik Karlsson, a postdoc from Sweden, that resulted in this, which for a little while was a record then in the efficiency of any of the LC columns. We have about 250,000 plates in 33 minutes. It's at some of the festive occasions. There are Karl-Erik Karlsson and Jim Jorgensen. These are the type of columns that are used today in the practice of proteomics, metabolomics, you name it. And this is the kinds of performance that, of course, gradually over the years has been improved. So there's really, and each of those things goes into the mass spectrometer. Each of them can be subjected to the GC, to MS type of routines. So this is the scientific lineage, and I'm very pleased that I could cause some of it. Milton Lee, who's produced his own, I was the graduate student here. Jim Jorgensen has been pretty busy in also producing the academicians and placing them at the universities. Most notably dear to my heart is Bob Kennedy at the University of Michigan, who by himself now has his own little lineage than the people who are at the university and receive tenure in academia. These are some of the others. Vicky McGuffin. I was visiting Brazil, and you know, there is a whole bunch of people who want to have a photograph with me. And I said, okay, I'll make a photograph with you and all that. And he said, you know, you are our scientific great father. So I was very pleased by that. Bill Alley, who recently moved to the University of Texas, and then some people who ended up being faculty members overseas, so they will have their own students. And needless to say, also the postdocs. I mean, I think Susan Olisic is in the audience. She was with us for some period of time in these interesting times during the 1980s. The international symposia, similarly, the story that Bill Heinemann was saying us play considerable role here. And so, again, due to the considerations of time, I won't go into that. Short courses that we were teaching then over a period of time, then, you know, also there were thousands of students that were subjected to that from industry and all this. Progress during the 90s and beyond, we finally got into with the advent of electrospray, turned LC-MS into reality. Implications for science are enormous. Chip-based separation techniques, what you can do in capillary, translates reasonably well into the chip. Ultra-high performance LC, and Jorgensen again is sort of was instrumental in starting off that field. Monolithic columns are available, competing with particle pack columns. We will see who wins in the long run. All kinds of sophisticated forms of mass spectrometry in combination with high-resolution separations are important in all these omics, proteomics, glycomics, lipidomics, metabolomics, and all the things which are actually dear to my heart. And not to miss to mention that also there is a GCGC, just like Gary mentioned. Yes, there is such a thing. And actually is followed by a number of people, particularly in the industry. Just very quickly, some examples of sort of materializing what sort of thing we were only dreaming about in 70s and 80s. This is a separation of polysaccharides that have been labeled with fluorophore, then subjected to laser-induced fluorescence detection. Monolithic columns, so one of these approaches. I won't talk about CEC, capillary electrochromatography, may one day again be important. Going to smaller dimension, again, chips, and they are commercial chips, and then there are also chips that have been designed by some clever people, like Steve Jacobson here, is collaborating with us. This is the device which has about 800,000 theoretical plates in it, and it's we use it for actually detection of the component where we feel might be biomarkers in the ovarian cancer and other type of cancers. And so this is more of the recent work. In case of the combination of ultra-high pressure with the small diameter, the same idea like there was with Jorgensen added about electrophoresis. The heat of friction is sort of negligible or very small, and therefore you can combine very high pressures then with HPLC, and this UPLC or so has been taken seriously by industry and by academic laboratories alike now, and that's the idea behind smaller the particle the better. And we more recently feel like if the particles are designed in such a way that there is actually a flow through the particles under the conditions of very, very small particles. This is in collaboration with my junior colleague Sarah Skrabelak at Indiana University. This may be yet another advance, and of course this last slide or so is from collaboration that we have with Jim Jorgensen again. So this is like, you know, a circle coming together or so of putting this under the best possible chromatographic condition. So in a retrospect, the small columns were initially met with skepticism, even adversity, I would say, but the last 2 decades have shown tremendous progress. Capillary LC&C have all these different applications, starting with genomic, proteomics, glycomics, etc. The human genome projects was accelerated by multiplex CE quite significantly. There's the whole club now of people who are able to study the individual cells or the small biological samples. I think Norm Dovicki is one of the examples. Of those undertakings. He's at this symposium, I noticed. And so that's basically what I wanted to tell you today. It's a little bit different. I see every lecture, I learn a great deal from previous speakers. Every lecture had a very different orientation. And so I hope I could add to it. Thank you.